time. So on the x-axis, we have year, and on the y-axis, we have abundance estimates. And you can see that there's this pattern in abundance. It's a decreasing pattern, unfortunately, which is true for many animals on the planet right now. And then there's, data points, and then there's error bars around each one of those data points. And then this line is fit to the pattern in the data. You can see there's this one weird low population in 1986, but overall the trend is shown there. So that's the same number, that's the number of Western gulls in a particular place at a particular time. And in this case, per year is the time frame. Okay. All right. So populations are characterized by several different features. So one is density. And so density is the number of animals occupying a defined unit of space. So this is the number of animals per area. So 50 spotted turtles per hectare or something like that, right? So you have a number of individuals in abundance that's divided by the unit of space that you're talking about. So that's density. This is actually a really useful metric for characterizing a population because it allows you to compare among different populations. So if you just said there's 50 turtles in an area and then somebody else caught 25 turtles in an area, you don't know how comparable those are if you don't account for the size of the area, right? So density is a really good way to scale your abundance estimate and allows you to compare those abundance estimates across different populations. Age structure is the ratio of one age class to another. So you might have hatchlings, number of hatchlings compared to number of juveniles compared to number of adults or something like that. So this is an age structure. It's a ratio of the different age categories you might create, you might define in a population depending on what you're trying to study. The sex ratio is the proportion of one sex to another, so the number of adult females to the number of adult males. This ratio is typically 1-to-1 in a healthy population, but there's definitely variation on that theme. And then birth rates and immigration are features that add to a population. So immigration is individuals moving in. Birth rates, of course, is what it says, how many are born. And then, of course, death rates and emigration, which is individuals leaving. Those are features that will shrink a population, make a population get smaller. And so then the difference between these gains, births, for example, and the losses, like death rate, then that gives you the growth rate of the population, right? So the differences between those gains and losses is what gives you the growth rate of a population, okay? So, no species is evenly distributed across its entire geographic range. Like you think about a range map for a particular species of animal. And so it's not evenly distributed throughout that range. The species is going to have higher density in some areas, lower densities in other areas. And also the species often occurs in sort of patches throughout that distribution, right? Distribution maps tend to shade in a whole big area and just say the animal occurs across this range, but it doesn't occur consistently across that range, right? So there's gaps that have no individuals. So there might be some intervening bad habitat, for example, that separates out individuals. So this idea that that it's not all continuously distributed within a geographic range, this leads to an idea called spatial dispersion, which is what we're going to talk about today. And so spatial dispersion, this is how individuals are dispersed or distributed across the space, so across the environment, across the landscape. So how are they dispersed across that space? And there's three general patterns of spatial dispersion. There's uniform, random, and clumped. Okay, so individuals might be distributed just sort of randomly all over the place. They might be equally spaced and uniform, or they might be in clumps. So we'll go through each one of these three with a quick definition. So a uniform distribution is the spacing of individuals that's more evenly than expected by chance. Okay, so they're more equally distant from each other than just random chance would predict. And so these patterns, when you have this equal spacing, this usually tells you something's going on between the individuals. So this usually means there's some kind of competition, some kind of intra-specific, some kind of interaction between individuals in that population that keeps them separated at these sort of equidistance sort of things. So, this kind of pattern, you might think, you see it maybe in something that's territorial, right? So if you have an animal that is territorial during its breeding season, in an area of relatively sort of equal habitat quality, then you might expect them to keep sort of, equal distances among each of those breeding sites. And so we have an example of that shown here using golden eagles during their breeding time of year. So the dark green on the outside of this is, not very good habitat. So the paler green is indicating the good breeding habitat for these golden eagles. And if you look at the sort of distribution of groups of sites belonging to one pair and the breeding, you can see that for the most part, these groups are actually fairly equidistant from each other. This is quite a uniform distribution of eagle pairs in these breeding territories. And so this is an example of that idea, because when they're in breeding, they're in territories and they're defending those spaces, which tends to lead to this equidistant distribution between individuals in the population. Okay. The second type of dispersion or distribution of individuals across the landscape is random. And so distribution is considered random if the position of each individual is independent of the others. They're not related to each other. There's no interaction. So the occupation of any spot on the landscape is equally likely. Okay. So random distribution is actually quite rare in natural systems, and partly because it can only occur when the environment is uniform across the board, right? Resources have to be equally available throughout the year, and interactions among individuals in the population have to have, there has to be no interactions that result in attraction or avoidance type behaviors, right? So there's no competition happening there. So again, this kind of random distribution is pretty rare, because you have to have a uniform environment where resources are equally available all the time. And you also have to have no interaction among members of the population. So there's no patterns of attraction or avoidance happening. So there are some data to suggest that spiders that live on the forest floor might actually follow this kind of randomized pattern. But again, it's pretty rare in nature, in natural systems. Okay, and then the third is clumped. And this is the most common type of spatial distribution. So this pattern results from responses by animals to differences in their habitats. And so there could be daily or seasonal weather and environmental patterns and changes. There could be changes in reproductive patterns and also social behavior. So you can see why this might, when you account for all those things, why this clumped pattern is probably the more common one in animals. Individuals might be drawn together in an area because of some common food resource or water or shelter, right? So an example here of a clump distribution are prairie chickens is an example, because they congregate for a communal courtship. They do a lekking arena, so they gather for that courtship. So there's a behavioral reason. And then another example is elk, for example, will gather together and have some social organization happening. Of course, wolves do that as well. So, this clumped type of dispersion is the most common in natural systems. There are two general ways that we can study spatial variation, two approaches to studying this variation in spatial dispersion. And so the first one is landscape ecology. And so this is determining how variation in certain aspects of physical structure of the environment, like topography, and then also living components like vegetative structure in an area, are important in terms of the animal distribution and the abundance and the diversity of different animals that might be there, right? So this is really, like, as it says, a landscape approach, right? And then there's a metapopulation approach. And this is the study of species when you view them as spatially structured groups of local breeding populations. So you'll have these local breeding populations, and you can have individuals that move between those local breeding populations. And that this is going to have, this is going to have an impact, of course, on what's happening to the population. So again, the species is spatially structured groups of local breeding populations, and then you're going to have movement of individuals among those little local populations. And we're going to focus on this view, this metapopulation approach, because it's more clearly sort of relates to how populations work. And today we're talking about populations, and also it's less like computer intensive and such. Like landscape ecology, as I said, is a really big scale. And so when you're using landscape ecology, it's lots of GIS and that type of modeling that needs to be done. So we're going to talk about the metapopulation approach today, and it applies to a lot of different animals, so it makes sense. Okay, so this metapopulation view of species, and here, this is how this works. So the environment consists of patches, of habitat patches. So these are areas of habitat that contain the requirements for persistence of the species, okay? So we have this patchy these patchy habitats, and each patch of habitat is separated by other areas of unsuitable habitat. So you have sort of this bad stuff in the intervening space, and then you have these patches of good quality habitat. Okay, so you have that happening on the landscape. And then each habitat patch has a population, a local population, which is called a deme. Okay, so each habitat patch then has a deme or a local population. And so this is a population of individuals of the species that occur within that same patch, and they interact the most with each other. So they reproduce the most with each other. And so since the habitat patches are separated by this unsuitable habitat, most of the individuals are going to then stay within their own deme, right? They're going to stay on their own patch, and they're going to hang out with their individuals in their own local population, as I said, and mostly interact with them and breed with them. But there's expected to be some level of dispersal that's happening between those patches. So there's going to be some level of dispersal between the patches, among the patches. And this dispersal is movement of individuals from their original deme to another deme. So there's connectivity. This is an important piece of it. Okay, this dispersal means there's some level of connectivity. Individuals can move from one patch to another, so join another deme. Okay, some or all of the demes and in the population are subject to extinction. So local extinction, because we're talking about just extinction in a patch. We're not talking about the entire metapopulation going extinct, but we're talking about the possibility that these little demes, these sort of subpopulations, are subject to local extinction. So one of those can go extinct, but other patches may remain occupied, right? So some or all of the demes in the population are subject to local extinction, so dying out. Then that leaves a habitat patch vacant, right? So there's a suitable habitat patch, but then there's nobody living in it, right? For some period of time, can last for several generations. And so this idea that you can have a patch that's unoccupied leads to this idea of recolonization. So patches can be recolonized by demes. And so this means that you can have individuals of the species that disperse into the vacant patch, right? And so you can have this winking in and out of these local extinctions and recolonization events, okay? And so metapopulations can reach or approach a sort of equilibrium number of individuals. So that's, a balance between this local extinction and recolonization idea, right? So it's, as I said, it's going to be sort of a winking in and out of individuals. But overall, of the whole metapopulation, there's some stability, but again, there's this sort of movement around and these local extinctions and recolonizations that are happening over time. Okay, so here's how it might look as a graphic to explain that, right? So you've got these patches of varying size. So here's a big patch and here's a little patch over here. These circles represent the patches. And each one of these patches has a local population of animals or a deme, okay? So there's a deme here, a deme here, a deme here, and a deme here, okay? And the arrows are indicating dispersal events that can happen between demes. So the individuals from the orange deme can move to the yellow deme and then back again. But you have these intervening patches of poor habitat. It looks like the habitat has a barrier between this blue patch and this green patch. So there's no individuals dispersing there. Okay, so as an example. So what you have, though, what this shows, this metapopulation system, is this interconnected populations of a particular species. So these interconnected demes, these subpopulations, okay? And again, this little subpopulation, this deme may go extinct, but then can be recolonized from this patch over here, okay? So one thing that's important to talk about here, and it's a distinction we need to make, is between dispersal and migration, okay? So dispersal, usually we're talking about unidirectional, one direction. So individuals from the orange patch can go to the blue patch, and they stay at the blue patch when they get there, okay? So it's unidirectional, not cyclical. But then there's this migration, which is a cyclical or like a seasonal movements, and it's repeated on certain timescales. Like individuals will make a migration to nesting sites, and they'll nest and then they'll come back. Okay, so that happens every year, and it's a migration. So that's not a dispersal; it's not a permanent leaving, right? It's a migration there and back. Okay, so that's an important distinction to make because here we're talking about dispersal, one-way movements. This metapopulation approach has been applied to species that fit this sort of structure of demes within habitats. And the demes have to be relatively independent from each other. So as I said, most of the individuals within a deem are interacting mostly with each other. And this idea then, of course, requires the habitat to be in these patches. So there has to be some kind of fragmentation almost to the habitat. This can be natural fragmentation, a natural mosaic, right, sort of habitats. And so this idea fits pretty well for lots of animal species. Examples like there's frogs and butterflies and small mammals have been shown to fit this. An example in my own research are spotted turtles, which tend to occur in these metapopulation sort of systems where they actually have breeding aggregations in the spring, and all the individuals within that breeding deem, are interacting with each other. So it fits this metapopulation sort of system in that there's this overall population, but then there are these separate breeding deems that spotted turtles use. Other species, they might look like they're occurring in habitat patches, but they have so much movement among those patches, maybe because the patches are really close to each other, or the animals have an incredible dispersal ability, that instead they sort of have, it's more like a really big population that sort of has patchy parts to it. But because there's so much dispersal between, then we don't really call that a true metapopulation system. So species might occur in habitat patches, but there's so much dispersal among those patches that basically you just have this sort of patchy population as opposed to these more discrete patches with deems in them. And so again, this can be, this dispersal, flying species. You think about birds, right? They can move from one patch to another so easily because of their ability to fly. So red-winged blackbirds are an example of this patchy sort of population as opposed to a true metapopulation. So, red-winged blackbirds occur in patches of marsh habitat, but there's a lot of movement between those marshes because they can fly. So it's not really quite, a metapopulation sort of system. A terrestrial barrier is no barrier to them because they can just fly over it, right? Okay, so another piece of this, so I've already sort of mentioned these terms of recolonization and local extinction. So to understand metapopulations, we have to understand what factors are going to affect these two processes then, these processes of recolonization and local extinction. Okay, so clearly landscape over which there's clearly going to be some barriers in the landscape, right? This is sort of the definition we've made. When we have these habitat patches, there's barriers between those patches. And so obviously then the landscape, which is going to have barriers to movement and the ability of a species to disperse, these are both going to obviously really affect colonization, right? Recolonization. How bad is the barrier and how well does that species disperse? So those are going to be important things to determine recolonization. In addition, recolonization is also affected by distance, right? So the distance between patches. So distance from a currently existing deem to the empty patches, right? So it's going to, if the patches that went extinct, locally extinct, is really close to another large patch, then probably the dispersal will come from that closest larger patch, occupied patch, right? So distance. Size is also going to matter. So the size of the currently existing deem. So, only so many individuals can fit within a habitat patch. And this comes to an idea we're going to talk about in a couple of lectures called carrying capacity of the habitat, right? That's how many individuals in a population can fit in that habitat given the resources that are there. So only so many individuals can fit into a patch because of the resources. So the size of the deem is also going to matter in terms of recolonization and individuals leaving. And then, of course, the dispersal behavior itself, right? Whether or not the animal in question can fly or it has to crawl or whatever, right? So those things are all going to affect recolonization. So we're going to talk about dispersal behavior then here for a little bit, just a little bit. So there are costs and benefits, right? And we talk about this sort of cost-benefit analysis a lot when we're talking about animal life histories and animal ecology in general. So the costs to dispersal is that it could be risky, right? You might not find a new habitat patch. So you try to leave your habitat patch and join another deem, but what if you never find it, right? Because it's new territory that you've never been to. So you might not find that new habitat. And then, of course, you're vulnerable to predation en route to that new habitat because you don't know where you are. You don't know where the shelters are. You don't know what risks are out there, right? So it's risky to disperse. But then there's benefits too, right? So you might go locate some mates. You might avoid competition in your current deem, which maybe is getting kind of crowded. You might actually find better habitat once you get there. And it also can avoid inbreeding, right? If most of the individuals within a deme are reproducing mostly with each other, and you don't want to interbreed with relatives, right? So you can avoid inbreeding by moving out. Okay, so there are different ways we can study dispersal. There are direct estimates, and then there are also indirect ways to study dispersal. So direct estimates can be obtained through methods like observation studies, right? Doing surveys, observation studies, doing trapping studies, like marker capture studies, right? Where you go out, and you know how to do this because we haven't done the marker capture lab yet. So you're going to learn about this, but this is where you individually mark individuals the first time you catch them and you let them go, and then you do a recapture and you can identify them, and then you can use that information to look at how individuals are moving, right? Because they're individually identified. And another way to study dispersal directly is to use VHF radiotelemetry, which you are going to do in your next lab after the break, and also GPS tracking. So just to say a little side tangent about this, you are going to learn about this in lab, but I'm going to give you a bit of a brief overview of what this is. So radiotelemetry, it works on the idea that you attach a radio transmitter to an animal. So here's a waterfowl of some species, has a radio transmitter attached to it. And you can see the antenna off the back. And then each one of these radio transmitters has its own unique frequency, just like a radio station. And then you use a receiver with an antenna, and you can dial in the frequency, the radio station of the animal you're interested in, and then you can follow its exact movements. So I have some examples. Radiotelemetry is what we use in all of the research in my lab. So I have some examples. So here's a small radio transmitter. You can see this yellow here. This is tape, because inside of that, this is a used radio. It came off of a turtle from a study that we just completed. The tape is actually holding a magnet onto the radio transmitter. So I'll pull that off. So there's a magnet. I hope you guys can see that. There's a magnet in there. And that magnet is there because there's a switch inside the radio transmitter that turns it on and off. So the magnet turns the transmitter off so that the battery life is not being wasted. And so you can see the antenna on the back. This glob of glue-looking stuff around the outside, that is the epoxy that we use to glue the radio transmitter to the shell of the turtle. And then the radio transmitter itself you can see a circle here and a sort of rectangle sticking off of it. So that's the radio transmitter itself. And then here's another example of a larger radio transmitter. And again, there's a magnet on it here to keep it switched off. You can see the main part of the radio there. And then around the outside here again is the epoxy that we use to glue it onto the turtle's shell. And then finally, I have an example here to show you what it looks like on a shell. This is Eddie. Eddie is a snapping turtle who was part of a study that we did. So here's the radio transmitter glued to Eddie's shell. And you can see the antenna actually is broken off of this particular radio. So the wire of the antenna broke off. So the sort of an interesting story about Eddie, just to show on the side. So Eddie obviously is deceased. Eddie's a snapping turtle who we radio track. So for those of you that are in the Sudbury area, you've probably noticed the fencing along the side of Highway 69. As you come up to town, there's the big chain link fencing, and then there's black fabric fencing along the bottom of that. And then that is actually part of a road mortality mitigation study that one of my students did. James Baxter Gilbert was the graduate student. And so what we did was we monitored the use of the fencing, and then there were eco passages under the road as well to try and guide the turtles safely across the road so they wouldn't get hit by cars anymore. So one of the things we did was outfit Blanding's turtles and snapping turtles with radio transmitters around the highway construction zone so we could monitor the dispersal and also the migration. So we were doing both of turtles around the highway and to look at how they interacted with the highway and did they use the eco passages that were built under the road. So the reason Eddie's sort of interesting is that she was radio tracked throughout the study, and then she disappeared from study, and then we never found her again. So obviously she died, but we don't know exactly when she died. That research was done quite a few years ago now. But this past summer, a man was out with his kids canoeing in our study area, and he found this shell up on the side of the lake. And he looked at the radio transmitter, and there's actually a window here that has some identification information. So that man contacted the Ministry of Natural Resources, and then who then phoned me to say, hey, we found one of your turtles with its radio. The radio doesn't work anymore. It's long dead. But it was nice to be able to recover the turtle and then now be able to use it for a teaching example. So that's Eddie. Eddie is named for the mascot for Iron Maiden because I love heavy metal music and my students know that. So the turtle's frequency is 666, which is the number of the beast, which is a song by Iron Maiden. So there you go, a small side tangent that Dr. Litzkis really likes heavy metal music. Okay. Oh boy, there's a comment. I don't know if it's about that or not. Okay. So that's direct estimates, and then indirect estimates of studying dispersal can be done by using genetics. So dispersal, immigration and emigration from one population to go breed in another is a form of gene flow happens, right? And we talked about forms of evolution, right? We talked about natural selection, but we talked about evolution being changes in the genetic composition in a population. So when you have individuals moving between demes, dispersing between demes, they bring their genes with them. So you have gene flow happening. So gene flow results in genetic variation within populations, right? You have gene mixing. So when you have gene flow and dispersal, then you're going to have this melting pot of genes, right? If there's a lot of dispersal happening among demes, you're going to have lots of gene flow. And so you're going to end up having high genetic variation, but not much difference in the genetics among the demes in those patches, right? However, if you have low gene flow, low dispersal, then you're going to end up seeing these demes that are very genetically distinct from each other. And I have a slide, the next slide tells this a little bit more. But so you can get information about gene flow and therefore dispersal, right, by looking at the genetics of these demes in these habitat patches. So again, we can, when you have little gene flow, you end up with genetic drift, right, which is fixation of alleles. So over time, by chance events, some of the alleles disappear out of a population. And so you end up with this very limited genetic diversity and this fixation of alleles, and we call that genetic drift. Okay, so there's various methods that have been used to study the genetics of populations. There's old school stuff and there's more modern stuff. So one of the older methods that was commonly used is this gel electrophoresis. And this studies proteins, which of course are coded by DNA, by genes, right? So the variation in these proteins is going to reflect genetic variation. And so electrophoresis uses electric fields to separate protein molecules that differ in charge across this starch gel. So you get slightly variant forms of the same protein or enzyme, and they're going to differ in charge, and so they're going to separate across this gel, and so then you can get information from that about genetic variation. More recently, of course, with rapid advances in molecular technology, you can just simply sequence the DNA, right? And that happens a lot more now, just straight up sequence the DNA. And then there's other things like restriction fragment length polymorphisms, and then also microsatellites, which are small random repeats, so small tandem repeats, of DNA. They're usually about 1 to 4 base pairs in length, and then you look at how much they repeat, and you can use that also to look at genetic variation. So there's, these days there's lots of genetic tools for this. Okay, so this is the graphic I was explaining just a second ago there. So when you're using genetic tools to look at whether or not dispersal is happening. So if you have dispersal, right, that means there's going to be lots of gene flow. So lots of individuals moving, right, between demes. And so then if these diagrams represent those demes, so what we have here is the V with the arrow up, that means high genetic variation, right? So if you have lots of dispersal happening among these demes, you're going to have lots of gene flow. So you're going to have high genetic variation. And you're going to end up with each deme looking quite similarly in a genetic context, right? So I have them all shown as circles to show that they're quite similar because there's lots of gene flow. So you have high genetic variation within demes, that's the V with the up arrow. And then the demes are going to be similar to each other, genetically speaking. So that's why I've represented them as circles. So if you detect these genetic attributes in a population, then you can conclude, if you detect this high genetic variability and not much difference among demes, you can conclude there's been a lot of dispersal. On the other hand, if you have a no dispersal situation, you're going to have that genetic drift, that fixation of alleles. And so within each deme, you're going to have low genetic variation. And each deme is going to be distinct from each other because you don't have individuals moving in between them, right? There's no dispersal. So they end up with fixation of alleles. So you have low genetic variation, and you also have distinction now among the demes. So I show this by a circle, a triangle, and a square to show that the genetics are different. So if you detect that when you're doing your genetic analysis, you can say, there's no dispersal happening or limited dispersal happening in this particular metapopulation. So some examples of studying dispersal for which genetic methods have been used include, for example, catfish studies. So in this case, different drainages had different populations with genetic difference. They were genetically different from each other. And that makes sense, right? Because catfish can't get up and walk between drainages. So you're going to have this distinction. So there's low gene flow, right, between those drainages. So there's low dispersal. If you look at pocket gophers, for example, there can be some genetic differences between the neighboring fields. And there are larger genetic differences between more distant areas, indicating that there's some gene flow locally, but it doesn't go very far, right? There's low dispersal at the more distant fields. And so this is an idea that's called isolation by distance, right? So the furthest away deme is going to be quite different than the more closely distanced demes to each other, okay? Red-winged blackbirds and song sparrows. So I kind of gave this example when I just talked about metapopulation structure. Like they fly, right? So they can move around. So for these guys, the populations, across the country have high genetic variation, but the individuals are genetically similar to each other. The populations are similar to each other, which means there's high dispersal, there's high gene flow, right? And that makes sense because this is an organism that can move around a lot, right? So most populations have high genetic variation and are genetically similar across the range. There's lots of gene flow. We're going to do a lecture on population genetics later on in the course. One little sort of side piece I just want to mention here is that there's dispersal and recolonization are not necessarily interchangeable. They're not the same thing, right? We can't just consider them to be the same thing. So you can have a species that could have high dispersal abilities, but actually low recolonization. So if the main benefit of dispersing was like to go find mates, for example, so that the dispersal went primarily to areas that already had members of the same species, then you're not going to have recolonization because there's already individuals there, right? So again, this might be an animal that can fly, like this butterfly, right? So it has high dispersal capabilities, but it's not the same as recolonization, because if the reason it's dispersing is to go find mates, that means it's going somewhere where the species does already exist, right? So it's not a vacant patch, it's a patch that's occupied. Okay. Another situation where you might have like high dispersal but low recolonization if all the dispersers are the same sex. So in many mammals, for example, it's primarily the males that disperse. So I show this red fox here is just an example of that males are the ones that disperse out. And this can help avoid inbreeding, but it's not going to promote recolonization, because if all males go and occupy a new patch, you're not going to have successful population because you can't have males reproducing just with each other. You need females there too, right? So if all dispersers are the same sex, then you're not going to have recolonization happening. Okay, so also to understand this metapopulation idea, we need to understand this recolonization part and also this local extinction part. So demes, basically by definition, are small populations, right? We're talking about these small pockets. And I mentioned that there's this tendency for them, to wink out and then recolonize. So this local extinction events tends to happen in small populations. And so then we ask the question, well, why are small populations more inclined to extinction, right? Why is there a higher probability? And there's 4 main reasons that small populations are more likely to go extinct. One is demographic stochasticity, environmental stochasticity, deterministic threats, and that loss of genetic variation that I've already mentioned. Okay, so first for demographic stochasticity, this has to do with random events associated with births and deaths. So they might lead to these types of random events can lead to a point in which no new individuals are born. And so we predict this to apply only in really small populations. And so what it's describing is random fluctuations in population size because of these sort of probabilistic events that are discrete of births and deaths. Because then you're going to end up with fluctuations because something can impact when the births are happening, for example. And so then maybe things will get sort of shifted off, and you're not going to have a bunch of births, but you're going to have a bunch of deaths. And so you're going to have a fluctuation, a decrease in that population. Okay, so again, it's this stochasticity around events associated with birth rates and death rates. And that, again, it goes with this idea that the birth and the death of each individual is a discrete probabilistic event that can vary. So this means that if all the individuals in the population are identical and have the same probabilities associated with birth and death, the precise timing and other details of births and deaths can vary randomly. So just the point to know is that when you have these random events that can happen that are dictating with birth rates and death rates, you can end up with fluctuating populations. And every time there's a decrease, there's an even greater chance of going extinct because small populations can just wake up then. Okay. Environmental stochasticity, this is random, unpredictable events in the environment now. So not having to do with birth rates and death rates, but having to do with the environment that can cause mortality events. So, for example, a severe weather event like a hurricane or something like that, right? This could lead to the loss of the entire population if you only have a few individuals in that population. It's, by chance alone, all of them being wiped out is pretty high when there's only a few to wipe out. And then there's deterministic threats. So these are non-random changes in the environment, and they tend to be directional changes. So the example here is basically the creation of unsuitable habitat because of some directional change. So a natural example is succession of habitat, right? So ecological succession. You have an open field that then turns to shrubs and then turns to forest, which then eventually becomes a mature forest, right? And different animals are going to be happier under those different scenarios, right? So that's a deterministic change. Of course, when you put the word threat on it, you can right away start thinking about human destruction of natural habitats as another deterministic directional change in a habitat. And then the last one there again is loss of genetic variation. And this occurs through genetic drift, which I've mentioned already, in inbreeding depression. So this is predicted to lead to extinction quite slowly, unlike an environmental stochastic event, which is an instant death of everything. So this happens a little more slowly. So it might be a little bit, maybe not quite as important as some of the other reasons for small populations to go extinct, but it's still going to have an impact. And so this, again, loss of genetic variation happens through this process of genetic drift that I mentioned, which is evolution that occurs because of changes in the alleles, right? And you tend to have, by chance, loss of alleles over time, which means limited genetic variation, right? And then fixation of alleles. So you just have low genetic variability in that group. And so over this means basically there's less potential for natural selection to act. So if something does change in the environment, there's such limited genetic variation in the gene pool that's there that there might not be sort of the genes to code for the phenotypes that would be able to tolerate that environmental change, right? Because there's so little genetic variation. Having a bunch of genetic variation means that if something changes, there's going to be a pool of individuals that have the genes that code for the phenotypes that survive okay and then reproduce fine in that environmental change, right? So again, genetic drift is a problem because of that. So you end up with limited genetic variation, so there's less a chance for a population to adapt to changes in the environment via natural selection. So you end up with a genetic bottleneck. You've probably heard that term before. Associated with genetic drift in small populations is also inbreeding depression. And these are decreases in production of surviving offspring because of reproduction between relatives, between close relatives. So when close relatives breed, that means the probability of recessive deleterious alleles are going to show up and be expressed, which can then lead to mortality, right? So when you have the probability of two individuals that are related to each other carrying a recessive deleterious gene, and then they breed with each other, then there's a greater probability that deleterious gene is going to show up in the offspring, right? So that's going to decrease survival. So and there's this idea that perhaps inbreeding might have less of an impact on populations that have historically gone through sort of these bottlenecks, because maybe all the deleterious genes were weeded out in that first event, and then maybe now the genes that remain are those that are okay. and the recessive harmful alleles have been weeded out already. So there may not be as much impact at present from inbreeding depression if this type of bottleneck has happened before. Okay. So the idea of environmental stochasticity that I mentioned, and also inbreeding depression, they can interact with each other to lead to something that's called an extinction vortex. So this is the idea that if the population becomes too small, and then you end up with some inbreeding, right? So relatives end up breeding with each other, and then you're going to have deleterious alleles, which means no survivorship, making the population even smaller, then making it even more vulnerable to having large fractions wiped out by some environmental event, some stochastic environmental event, which then results in smaller population with more inbreeding and so on and so on. And so you have this spiraling down. So this concept, it leads to this vortex, this spiraling down of population size. Again, because of this, the interacting effects of environmental stochasticity and inbreeding depression, leading to this extinction vortex. Okay, and you might have, I don't know if you've heard of this term, but vortex is actually the name of a software program that is for doing population viability analyses, and it's based on this concept. Okay, so this idea of extinction vortexes has led biologists to think about what is the smallest population that can sustain itself. So what is the smallest population, the minimum viable population size that's going to prevent an extinction vortex? And there's been a few rules that have been thrown around about this, but some have suggested that a population size of 50 individuals is considered to be the minimum viable population size. So the idea being that 50 individuals is enough to bounce back from a bottleneck. Another way that people have looked at minimum viable population is that it's estimated as the population size that's needed to ensure that 90 to 95% probability of survival from 100 to 1,000 years into the future. And this idea has been based on doing computer-based analyses of population viability analyses. So that software program, Vortex, that I mentioned, is a program that can do these kinds of calculations. Okay, so there's that idea as well. And then there's this other idea that, so I mentioned the idea that 50 is the minimum number of individuals that you need to bounce back from a bottleneck. And others have suggested that that doesn't mean that a population that's bigger than 50 is necessarily going to avoid an extinction vortex. And so folks have suggested that instead what you need is 500 individuals for long-term viability. Some have even said it's 200 to 250. But there's this idea that's called the 50-500 rule. And so that's this idea that, that you can bounce back from a bottleneck at 50, but if you have 500, that's better for long-term viability of the population. And in my personal opinion, these types of rules are far too simplistic because this doesn't account for the life history of the animal, the type of breeding system it has and stuff. So I think that these are sort of silly. Personally, I think this is a silly idea. And to me, this idea of doing a population viability analysis, which actually includes information about the life history of the animal you're talking about, are much better ways to determine what minimum viable population size is. I don't think you can apply a rule across all different animals. It just doesn't make sense to me. Okay, so local extinction depends on population size, as I already said, right? So small populations have a greater chance of going extinct. And it's based on those different things: demographic stochasticity, environmental stochasticity, et cetera. So there's also the impact of patch size, right? That habitat patch is also going to affect the deem size, right? Because of that carrying capacity idea. And so this is going to affect what happens in a metapopulation system. So metapopulations have been considered to fall into a continuum then. So there's at one end these classic metapopulations, and at the other end, this mainland island idea. So a classic metapopulation, this is the idea that all deems are small and all are equally likely to go extinct. And so that's sort of at one extreme of a continuum. They're all small and they're all equally likely to go extinct. And on the other end, we have this mainland island idea where some deems are very large and likely to persist, and then some deems are small. So the larger deems are surrounded by smaller deems that undergo local extinction, but there's always this larger deem in the middle to sort of rescue everything. And this mainland island idea is also related to an island, an idea called island biogeography, and I'm going to talk about that idea later in a lecture. But talking about this mainland island metapopulation idea, this has to do with variation in habitat quality. So, up to this point, we've been assuming that all areas that could have a deme are equal in quality, right? We're assuming that every one of those patches of habitat are equal quality, and probably that's not true in most natural systems. One particular difference in habitat is that there might be some areas of good habitat, right? You might have an area of good habitat where more individuals are born than die. So that means your population has a higher birth rate, B, than death rate. So B is bigger than D. Birth rate is bigger than death rate. So of course, that means the population is going to grow. If you go back to one of the earliest slides in the lecture, and I said that when birth rates, contributing to the population, you're going to have a population that grows. Population growth rate is examined by looking at the difference between birth rates and death rates, right? So you might have an area of good habitat, lots of individuals are born because there's lots of resources, and there's fewer that die, so you're going to have a population that grows. Okay, and then you might have some crappy habitat, some poor habitat, and now death rate is going to be bigger than birth rate. So now death rate is bigger than birth rate, and so the population is going to get smaller in these areas. Okay, so again, you can have these differences, and this is probably more realistic. So we're going to, if more are born than die, then we're going to suggest that this population in the good habitat grows, and if more die than are born, we're going to suggest that the demes in the poor health quality habitat are going to decline in size. They're going to die out. So these areas might remain relatively constant in population size, though, if you have individuals moving around, right? And so this idea of a metapopulation system is based on the idea of individuals can move sometimes, right? They can disperse between their patches and move between demes, and hence the gene flow. So population size overall, the whole metapopulation, might remain relatively constant if you have individuals that are moving. So individuals will move out of the good habitat. They're going to emigrate out because it gets pretty crowded. If your birth rate is bigger than your death rate and the population is growing, it's not going to be a lot of elbow room. And so individuals will move out of the good habitat and then move into the bad habitat. They're going to immigrate into the bad habitat because there's more space there. Although the habitat's kind of crappy, but there's more space. And so you're going to have this balancing of emigration and immigration. And so there are population models that look at all these things, these birth rates, these death rates, immigration and emigration rates. And they're called BIDE models for that birth, immigration, death, emigration rates. And so based on this idea of variation in habitat quality and this variation in birth and death rates, it leads to this idea of source and sink populations. So in a source population, this is where you have birth rates that are higher than death rates, right? The source population, birth rates are higher than death rates. So you have good quality habitat, right? We've said that in good quality habitat, this is what you expect to see. And so what's going to happen is you have a growing population, a high birth rate in this good habitat, and individuals are going to leave because it's going to get crowded, right? So you're going to have emigration is higher than immigration in this case, right? So higher emigration than immigration. More are going to leave than arrive, okay? So again, this is predicted to grow, to be in areas of good quality habitat, but when it gets crowded, individuals will leave. And so the population is growing, right, in these sources. As opposed to sinks, here we have a case where the death rate is higher and the birth rates are lower, and you're going to have more immigration. So more individuals are going to be arriving. So this is what you expect in those poor habitat patches, poor quality habitat patches. So individuals immigrate into them when the sources become crowded, right? And so this is what maintains these sink populations are because of the immigration that's coming from the source, right? So even though the habitat's poor, it's just getting so crowded in the source population that individuals still leave that source and go to the sinks. But the sinks, it's not a great spot to be, right? Because the habitat is poor. So you can think about this, these sink populations, the name is that it's going down the sink, it's spiraling down the sink because the individuals, they go down the drain, so to speak, because the death rate is higher here. But again, these sink population are maintained because of the source population, individuals leaving the source and going to the sink. So we can, one of the ways to distinguish, if you have a metapopulation system and you're looking at the demes and your patches and you want to know, is it a source population or a sink population? The way you can do this is to keep track of the birth rates and the death rates, and also immigration and emigration rates from into and from those demes, those patches, right? And you can do this by using something called life tables, which allow you to keep track of individuals. And next lecture, we're going to talk about life tables. It's an important demographic concept in animal ecology. So we'll talk about that next time. But one thing that's important to think about is this existence of these source and sink populations must be considered when you're setting aside nature reserves for species at risk, right? Like, what if you set up a reserve and almost everything in there was a sink, that is poor habitat quality patches? And so then, you're wasting your time and energy. It's crucial to set up nature reserves that maintain source populations where the birth rates are higher in good quality habitat. If source populations are not maintained and you set up a reserve where there's just, like I said, a bunch of sinks, then the species we want to preserve is just going to die out because there's not enough resources in those poor quality habitats. So something very much to consider when setting aside nature preserves for a particular species. So here's kind of how it looks as a picture, again, of a metapopulation system with sources and sinks. So in this example, we have a source population on this patch, and we have another source population on this patch. So again, these are high quality habitats where the birth rates are greater than the death rates, and where emigration is greater than immigration, right? More are leaving. And then you have these sink populations, these little guys around the periphery, right, that are in poor quality habitats. And in these places now, the death rate is higher, right? But this little sink is maintained because individuals leave this source and go there. So the arrow's not pointing the other way in this particular case of this one. So the animals sort of move from this source, this healthy habitat source, because it's getting crowded though, because the birth rates are higher. And so they move over here, but then you have a higher death rate. And so this population will sort of decline, but it just keeps in existence because individuals keep moving over there from the source. Okay, so given this bit of a review, so we've got lots of time actually. So to go over some of the things we then talked about today. So we talked about some population characteristics. So these types of things we talked about here, right, were density. So density, again, is a measure of population abundance, but it's a really useful one because it standardizes against the space over which that population exists. And so then it allows you to do comparisons among populations of a given species. So it's really useful to use this density estimate. Another thing we talked about were age class ratios and sex ratios are important characteristics that describe populations as well. And then, of course, we talked about birth rates and death rates and population growth. And we'll talk more about that again, population growth especially. We're going to talk about quite a bit more coming up. All right. And then we talked about spatial dispersion. So we talked about uniform, and this tends to happen when there's some interaction happening among individuals. So the classic example here is intra-specific competition in terms of territoriality at nesting sites, for example, or breeding sites, which causes individuals to be equally distant from each other than would be expected by chance alone, right? There's spacing. There's equal spacing among them because they're defending territories, for example. Random is any particular spot on the landscape is as equally likely as another spot on the landscape. Pretty rare in nature because it assumes that everything is the same on the landscape, which is not always the case. And then clumped, which of course is the more common one, and this is where individuals tend to be clumped together around certain resources, for example. And so you then have clumps of individuals in this area in the environment and another clump of individuals over here, and then there's gaps in between where you don't have as many individuals. Okay. And then we talked about the metapopulation system. So we spent a lot of time talking about that idea today. And the basic premise here again is that individuals in a population across their geographic range of that species are not equally distributed right across that whole space. Instead, we have habitat patches, and intervening are patches of habitat or pieces of habitat that are not good quality for that animal. There's barriers maybe or whatever it might be. But so you end up with these habitat patches. And within each of those habitat patches, you have a local population or a deme. And individuals within that deme are mostly interacting with each other, mostly reproducing with each other. But every now and then you have dispersal events that happen between those demes. Okay. But everything together, so all those demes in their patches, the whole thing together is the metapopulation, so the bigger population. And then these subpopulations or demes are what make up that bigger whole metapopulation system. Okay. We talked about the idea of recolonization and dispersal playing a role in how this system works. So the idea is that when you have some patches that, patches tend to have small populations, each deme tends to be pretty small. And so there's this tendency for these small populations to go extinct because of stochastic events in birth and death rates or stochastic environmental events. And then that makes them more susceptible to going extinct. And we can have these things called even extinction vortexes, which is a spiraling down of population size due to environmental stochasticity mixed with inbreeding that just leads to smaller and smaller populations in this feedback loop, right? This positive feedback loop into extinction. So what that means is if a deme goes extinct from a patch, there's an opportunity for recolonization to happen though, right? From an adjacent patch of habitat, individuals from that deme might go over and recolonize, okay? And so then again, what that means is that the overall metapopulation is going to be fairly constant in size, but you're going to have these winking in and out and moving about recolonization and local extinction events that are happening among those patches. Okay, and so I've already mentioned then this tendency for small populations to go extinct, primarily due to stochastic events. But there's also directional changes too that we talked about, deterministic change, like habitat succession or human destruction of habitats. And then related to this idea of recolonization and dispersal, immigration and emigration is this idea that not all habitats are created equal, right? So there's going to be different quality habitat patches. And so you're going to have some cases where you have sources where the birth rate is higher than the death rate. So the population is growing. But because the population is growing, it's going to cause some problems for elbow room. And so individuals will leave and they'll move over to sinks, which tend to occur in lower quality habitats. But those sinks, those demes are maintained because of individuals leaving these sources. They tend to be poor quality habitat and the death rate tends to be higher than the birth rate in those sinks. And so one thing to keep in mind in terms of conservation of species at risk is to consider the habitat patches that you set aside in a conservation reserve, for example, to make sure you have sources in there so that you're not just basically introducing animals to a site and then watching them disappear. And with that, I'm going to wish you all a really great Thanksgiving weekend and also a really great reading week. And I hope you all get a chance to get away from your screens for a little while, give your eyeballs and your brains a break from that screen stuff we've been doing so much of with all the teaching on Zoom. And with that, I will stop sharing my screen and we can chat about anything that you might want to chat about. Let's see. Does a beaver dam creating a new pond count as deterministic? That's a really great question, actually. And I would say yes, because it is a directional change in that habitat, right? And it doesn't mean that after some period of time, the beaver dam isn't, might get broken and then you'll have a return to another habitat, a less flooded habitat, for example. But I would say it is a deterministic event because it's a progressive change. And that's a great question, actually, Janine, because I have a particular experience of this in one of my metapopulations of spotted turtles that I've been studying. One of the breeding aggregation sites, one of the breeding demes, was occupied by beaver who then flooded it and the water level doubled, the water surface area doubled, and that caused the turtles to actually leave. All the spotted turtles left that breeding site and they no longer stay there because of that deterministic change in the habitat. Let's see, Emma said, do the transmitters most commonly come off when turtles shed their scutes, or do you normally have to physically remove them? And so that's a really good question too from Emma. So you can see that, well, you saw when I showed you, Eddie, that right? This radio transmitter is still glued on here. The epoxy is still there, and it's still quite well attached. And that's been, I'm trying to think how many years it's been. It's probably 6 years, maybe, that's been attached. But I don't know how long the turtle was dead before the canoeist found it this summer. But you can see that this, there's a scoot that's actually loose here. I don't know if you can see that, but so it depends on how the turtle sheds about whether or not the radio transmitter will be shut off. Most of the time, we retrieve our animals and we pop the radio transmitters off because we don't want them wandering around with radio transmitters on them. And so typically when we do that, this one shows it a little bit. I don't know if you can see it. You probably can't see it, but underneath this edge of the radio transmitter, we try to leave a little bit of you can say a notch in the epoxy so that we can just put like a Swiss Army knife and go pop it off. And it comes off quite easily. You can see, so this radio transmitter was attached to a painted turtle. And you can see this black right here. That's actually a little bit of the keratin, that layer that came off, the part that would shed if the turtle was shedding naturally. So it doesn't bleed, it doesn't do anything like that, it doesn't harm the turtle, but it might pull off, like this one did, a smidgen of the keratin layer that would naturally shed off. So we've certainly had the case where radio transmitters fall off, and it's super frustrating because they're just shed off or they're knocked off. Like, we try not to make them permanently mounted on there in case we can't re-find the turtle. But most of the time, our protocol is to recapture turtles and remove radios when the study is finished. Darwin says it kind of looks like a mini horseshoe crab. It really does look like a mini horseshoe crab. And that's because the epoxy that's around the outside that's on there. Are there any other questions? Anything anybody wants to talk about before the fall reading week? No? All right. If not, then I will wish you all a happy Thanksgiving and a good reading week. Take care, and I'll see you when we all get back.